The line between organism and machine is becoming increasingly blurred. We are standing at the edge of a new technological epoch, one defined not by silicon and steel alone, but by the unprecedented fusion of living biological matter with synthetic, man-made structures. This is the world of biohybrid robotics, a revolutionary field that promises to redefine our understanding of life, robotics, and the very nature of what it means to build. These living machines are no longer confined to the pages of speculative fiction. They are taking shape in laboratories across the globe, performing tasks from swimming and crawling to sensing and self-healing. This is the dawn of a new kind of creation, one that leverages the evolutionary genius of biology to animate the innovations of engineering. This journey toward biohybrid robotics is not a sudden leap, but a gradual convergence of multiple scientific disciplines. Its intellectual roots trace back to the mid-20th century with the rise of cybernetics, a field that studied the communication and control systems in animals and machines. Early pioneers like W. Gray Walter with his robotic tortoises Elsie and Elmer in the late 1940s demonstrated how simple electronic systems could produce complex, lifelike behaviors just by interacting with their environment. While these creations were purely mechanical, they planted the seed of an idea that the principles governing biological life could be replicated in artificial systems. For decades following that, the dominant paradigm was biomimicry, designing robots that imitate nature. Engineers drew inspiration from the efficient locomotion of snakes, the dynamic balance of legged animals, and the hydrodynamic grace of fish to build more effective machines. This led to incredible innovations, from snake-like robots capable of navigating rubble to hexapod robots that walk like insects. However, these were still fundamentally machines made of conventional materials, powered by batteries and motors. The true shift toward biohybridization, the direct integration of living tissue into robotic systems, began to gain momentum in the 21st century, fueled by parallel advances in tissue engineering, materials, science, and microfabrication. A pivotal moment occurred around 2012 when researchers demonstrated a micro-robot powered by insect muscle tissue, which moved faster than previous designs. This was a clear demonstration that biological muscle could serve as an efficient engine for a synthetic body. The field captured wider attention in 2014 when a team at the University of Illinois, led by pioneers like Rashid Bashir and Tahir Saif, created a tiny, soft biobot that could walk by harnessing the rhythmic contractions of rat heart muscle cells. This was a seminal achievement, proving that living cells could be cultured and integrated onto a flexible, 3D printed scaffold to produce directed motion. Just 2 years later, in 2016, a team at Harvard University, led by Kevin Kit Parker, unveiled another landmark creation, a small, autonomous stingray-shaped robot. This biohybrid stingray had a gold skeleton overlaid with a flexible polymer and was powered by a layer of approximately 200,000 living rat heart cells. Crucially, these cells were genetically engineered to be light-sensitive. By flashing blue light, the researchers could stimulate the cells to contract, causing the stingray's fins to undulate and propel it through a nutrient-rich liquid. By varying the frequency and location of the light pulses, they could even steer the robot, making it navigate a simple obstacle course. This project was a stunning showcase of how genetic engineering, tissue engineering, and soft robotics could converge to create a complex, controllable living machine. So, how exactly do you build one of these biohybrid robots? At its core, each one is a masterful combination of three fundamental elements: the biological actuator, the synthetic scaffold, and a control system. First, let's talk about the living engine, the biological actuator. This is the living part that generates force and creates movement. The most common actuators are muscle cells, prized for their natural ability to convert chemical energy from nutrients into mechanical work with remarkable efficiency, sometimes up to 50%. Researchers primarily use two types. The first are cardiomyocytes, or heart muscle cells. These are popular because they contract rhythmically and spontaneously, providing a constant, pulsing motion without needing continuous external stimulation. The walking biobots and the Harvard Stingray are prime examples. The downside, of course, is that this spontaneity makes them harder to control precisely. The second type are skeletal muscle cells. Unlike heart cells, skeletal muscles contract only when stimulated, typically by an electrical or optical signal. This provides a much higher degree of control, allowing for more complex and dynamic movements like starting, stopping, and turning on command. In January 2024, scientists in Japan demonstrated this potential by building a two-legged walking robot using lab-grown human skeletal muscle. To achieve even more sophisticated control, some researchers are integrating neurons or nerve cells to form neuromuscular junctions, mimicking the way a brain tells a muscle to move. In 2024, a team at Harvard Medical School created a butterfly-like swimming robot from human-derived neurons and muscle cells. The onboard neurons formed functional connections with the muscle tissue, allowing a programmable electronic brain to control the fin flaps wirelessly. This brain-to-motor frontier is a crucial step toward creating autonomous systems capable of adaptive control and even learning. And at the microscopic scale, single-celled organisms like bacteria, algae, and even sperm cells serve as powerful engines for microbots. These microbes are natural swimmers, using their tiny whip-like tails called flagella to propel themselves. Researchers can attach synthetic cargo, such as drug-filled nanoliposomes, and then use the bacteria's natural motility, often guided by external magnetic fields, to navigate complex biological terrains like the human bloodstream. Next, the biological engine needs a body to act upon. This is the synthetic skeleton, or scaffold. This scaffold provides structure and channels the force of the living cells into useful motion. The primary requirement for any scaffold is that it must be biocompatible, meaning it's not toxic to the living cells it supports. Many designs use soft, flexible polymers like hydrogels, which are water-rich polymer networks that closely mimic the natural environment in which cells thrive. Materials like gelatin methacryl oil, or Gel-MA, a modified form of gelatin derived from collagen, are widely used because they are biocompatible, biodegradable, and can be easily customized. Other synthetic polymers like PCl and PLA are also common, especially because their degradation rate can be controlled to match the device's intended lifespan. This is a huge advantage for applications inside the human body, as the robot can naturally decompose after completing its mission, leaving no harmful waste behind. A significant advancement has been the move from soft, viscoelastic materials toward linear elastic materials. These newer platforms behave more consistently across a wide range of movement frequencies, resulting in robots that generate larger, more reliable, and more powerful movements. Advanced manufacturing techniques are critical for creating the intricate architectures needed to house these cells. 3D bioprinting has been a game-changer. It allows researchers to build complex, three-dimensional scaffolds, layer by layer, often printing both the synthetic material and a bioink containing living cells and nutrients. This precision allows them to create specific structures that guide muscle tissue to grow in optimized patterns, as seen in the Harvard Stingray, where protein lines were micro-patterned to align the muscle cells for efficient contraction. Techniques like soft lithography, borrowed from the semiconductor industry, also allow for the creation of micro-scale features on soft materials, essential for building micro-actuators and tiny labs on a chip. Finally, to be a functional robot, it must be controllable. The most common method of control is external stimulation. Researchers can apply an electrical field across the muscle tissue to make it contract. This is straightforward, but can cause muscle fatigue and damage over time, if not carefully managed. A more advanced and precise technique is optogenetics, which involves genetically modifying the cells to respond to specific wavelengths of light. This offers highly precise, non-invasive control with high spatial resolution, allowing researchers to activate specific parts of the robot independently. The ultimate goal, however, is to create fully autonomous biohybrid robots with onboard sensors and control circuits. Researchers are exploring embedding sensors within the artificial muscles to provide feedback, allowing the robot to know what to do next. In a fascinating development just this year, scientists at Cornell University have even used the natural electrical signals from fungal mycelium to control a robot, harnessing a living system's innate ability to sense its environment. This theoretical promise is being realized through a growing menagerie of remarkable creations, illuminating a vast range of potential applications. Perhaps the most transformative applications are in medicine. Microbots, powered by bacteria, are being designed as intelligent drug delivery vehicles. Imagine loading these tiny robots with chemotherapy drugs and guiding them with magnetic fields directly to a tumor. By releasing their payload only at the target site, they could dramatically reduce the debilitating side effects of conventional chemo, which floods the entire body with toxic agents. Researchers have even engineered biohybrid microswimmers using red blood cell membranes to reduce the immune response, making them stealthier couriers within the body. Beyond that, the vision of nano-doctors performing surgery from within the body is slowly moving toward reality. Biohybrid robots, with their soft bodies and precise control, could one day navigate through blood vessels to perform microsurgeries, remove blood clots, or take biopsies without a single incision. Beyond medicine, biohybrid robots offer new tools for monitoring and restoring our ecosystems. Imagine biodegradable robots made from living cells that can patrol oceans, detecting toxins, neutralizing pollutants, or collecting microplastics. Researchers are exploring the use of bioengineered bacteria that can break down hazardous materials, like oil spills. In 2024, engineers at Caltech demonstrated that by equipping live jellyfish with small electronic devices, they could make the animals swim faster and more efficiently, turning them into highly effective data collectors for monitoring ocean temperature, salinity, and oxygen levels, key indicators of climate change. In the chaos after a disaster like an earthquake, biohybrids could also become critical search and rescue operatives. Researchers at institutions like Nanyang Technological University have developed systems for equipping insects, such as Madagascar hissing cockroaches, with tiny electronic backpacks. These cyborgs can be remotely controlled to navigate through rubble and crawl into crevices inaccessible to larger robots or human rescuers. Equipped with infrared sensors or microphones, they could detect human presence and wirelessly alert rescue teams. Scientists are even developing swarm navigation algorithms, allowing groups of these cyborg insects to work together, coordinated by a leader insect, to systematically search a disaster area, a task that leverages the insect's natural mobility while overcoming their individual unpredictability. No discussion of this field would be complete without mentioning xenobots. Unveiled in 2020 by scientists at the University of Vermont and Tufts University, these are not robots in the traditional sense, but entirely new, programmable organisms. They are created from the stem cells of the African clawed frog. A supercomputer designs a specific shape for a task, and scientists then assemble the frog's skin and heart muscle cells into that configuration. The results have been astonishing. Xenobots can move, push pellets, carry payloads, and even heal themselves when cut. Most remarkably, in 2021, the team discovered that xenobots could self-replicate. They do so in a manner never before seen in biology. Parent xenobots, shaped like Pac-Man, move around their petri dish, gathering loose stem cells into piles, which then self-assemble into new baby xenobots. While their immediate purpose is to help scientists understand the software of life, how cells cooperate to build complex bodies, their potential future applications are vast, from cleaning arteries to targeted drug delivery. Now, despite all this incredible progress, the path to a future populated by functional living machines is fraught with significant challenges. The living components are their greatest strength and also their greatest weakness. Muscle tissues are delicate and require a highly controlled environment with a constant supply of nutrients, a stable temperature, and proper hydration. This makes operating them outside of a laboratory setting extremely difficult. Their lifespan is also limited as biological tissues naturally degrade over time. Powering and controlling them is another major hurdle. Most current designs are either tethered for power or rely on external fields like light, which are hard to apply in complex, real-world scenarios. Developing robust, long-lasting, and fully integrated onboard power sources is a critical area of research. And so far, most of these robots are tiny, operating on the millimeter or centimeter scale. The forces they generate are minuscule. Scaling them up is a monumental challenge. Growing larger muscle tissues is a slow process that takes weeks, and it requires the development of vascular networks, akin to blood vessels, to deliver nutrients and remove waste from deep within the tissue, something tissue engineering is still striving to perfect. Finally, biological systems have inherent variability. Unlike mass-produced mechanical parts, no two pieces of cultured tissue are exactly alike. This makes it difficult to create robots with robust, repeatable, and predictable performance, a cornerstone of traditional engineering. As we venture deeper into creating entities that are part life and part machine, we also confront profound ethical, legal, and social questions that demand careful consideration. The most fundamental question revolves around the moral status of these creations. As they incorporate more complex neural tissues and exhibit more lifelike behaviors, we have to ask, could they develop the capacity to feel pain or achieve some form of sentience? If so, what are our moral responsibilities toward them? Treating them as mere machines could become ethically problematic. Releasing autonomous or self-replicating biohybrid robots into the environment also raises serious concerns. A biobot designed to clean oceans could be ingested by marine life, disrupting the food chain, or have other unforeseen and irreversible effects on natural ecosystems. Furthermore, like many advanced technologies, biohybrid robotics poses a risk of exacerbating social inequalities. If biohybrid enhancements offer superhuman capabilities, their high cost could create a new divide between the wealthy who can afford them and the rest of society. And the potential for dual-use applications, particularly in warfare, such as biohybrid drones or surveillance insects, necessitates robust governance to prevent misuse. There is a striking consensus among ethicists and many scientists in the field that these issues must be addressed now, not after the technology is mature. Despite thousands of research papers on biohybrid robotics, only a tiny fraction have explored the ethical implications. Projects like Biohybrid Futures are now working to create frameworks for responsible research and to engage the public and policymakers in a dialogue about the future we want to build with these living machines. So what does the future hold? The field of biohybrid robotics is still in its infancy. Today's creations are the equivalents of the first vacuum tube computers. The future trajectory points toward increasingly complex, autonomous, and capable living machines. Researchers are intensely focused on developing more sophisticated neural interfaces to create robots that can learn, adapt, and make decisions. They are working on engineering self-sustaining systems with built-in vascular networks and the ability to extract energy from their environment, freeing them from tethers. They are using AI to rapidly design new biohybrid architectures and to streamline the slow process of tissue cultivation. And they are working to fully realize the potential for biological regeneration, creating robots that can repair themselves from damage. In the long term, the vision extends to frontiers as diverse as space exploration, where resilient, self-sustaining biohybrid robots could explore alien worlds, and regenerative medicine, where the principles learned could allow us to build fully functional, patient-specific artificial hearts and other organs. The dawn of biohybrid robotics is more than just a technological revolution. It is a philosophical one. These living machines force us to confront the very definition of life and our role as creators. By merging the evolved wisdom of biology with the inventive power of engineering, we are not just building better robots, we are gaining a deeper understanding of life itself. The journey ahead will be complex and filled with challenges, but it promises a future where our machines are not just intelligent, but truly alive.